Hello again. So in this section, we're going to attempt to finish off the Alzheimer's disease part. So, and then in the next section, we'll do probably 2 videos of Parkinson's as well. Okay, so we're a bit worried that this amyloid cascade hypothesis isn't what's actually happening in patients. Are there other hypotheses? Well, I think it would be easier to say what hypotheses aren't there. I mean, there's lots of them. I'm not going to go through these in any kind of detail just because there are so many of them. We don't have the time for that. The amyloid cascade hypothesis is probably the one that's most widely known and used and understood. So I'm just going to briefly mention these. And I don't know if you've noticed, but I've got these on most of the slides. I've included references. So these are the DOI links. And so they should be permanent. So if you click on any of these links, that should take you to the reference that the information or the image on the slide are based on. I've usually got the links generally next to the information that comes from that reference, but it's not absolutely exact. Because I've included some things sometimes from things I know and just other references. So, but generally the references are some of where I've gotten the information from. And so if you're more interested in finding out more about, I've got the references in a lot of the different slides. So I've mostly tried to include references for everything. So here, this one is just particularly for the image, and the information is more from this other reference. But anyway, sorry, I shouldn't have gone so back so far. So please just, if you're interested and want to find out more about any of these things, please just follow the reference. Follow the link, and you can see the references. I do have some increased access to some journals because I'm still a member of staff at, I'm visiting sort of honorary member of staff at Newcastle University, so I have access to their journals as well, and I don't always keep track of which ones. So if you follow a link to a reference and you can't access it with Teesside access, just drop me an email and say, I'd really like to read this paper, and I'll just email you the PDF. Okay, so one of the other hypotheses is the mitochondrial cascade hypothesis, which involves mitochondria, as you would expect. There is the dual pathway hypothesis, and I think this one involves kind of amyloid and some other pathways. There's the metabolism hypothesis. This has to do with glucose metabolism in the brain. A cell cycle re-entry hypothesis. So, neurons aren't supposed to undergo any cell cycle because they stop once they become neurons. There's a vascular hypothesis, which has to do, obviously, with the vasculature and where exactly the plaques are and things like that. They're in certain places along the vasculature. There is the A-beta oligomer hypothesis. This is a hypothesis that, and this is something that people are thinking more and more, that actually it isn't the plaques that cause the problems in the patients. It's not the A-beta plaques that cause the problems, but it's the oligomers. So if you remember that one figure, the oligomers were like the first step in sort of the aggregation process and creating the plaques. And people have an idea that it's actually the oligomers that are toxic, and that our brains have developed the protective technique of making the plaques to keep the oligomers from being toxic, essentially. There's sorry for this misspelling, but there's a cholinergic and hypothesis, and then there's a cholinergic and oxidative stress hypothesis. There's a dendritic hypothesis, and there is the tau hypothesis. And of course, the NFTs being such a big part of the pathology, I'm going to go more, talk a little bit more about tau now. And then there is also the microglia inflammation hypothesis. So again, and just to let you know that generally these hypotheses here go with this reference. These two references, so this reference is kind of these hypotheses, and this reference has kind of these hypotheses. I think that's how I organize them on the screen. But as you can see, there are a lot of different hypotheses as to what exactly is going on in Alzheimer's disease. So, what about tau? You guys will have been told about the NFTs in Alzheimer's disease. They are caused by tau aggregation, well, not aggregation, but they're caused by the tau. And what is tau? Tau is a neuronal specific microtubule associated protein, and it's mainly found in the axons of the neurons. It's normally an unfolded soluble protein that interacts with tubulin to promote microtubule assembly and stability. So, microtubules are very important parts of cells that keep their structure. And they're also used for transport of various things along the neurons as well. So in AD, tau is abnormally hyperphosphorylated. So what does that mean? That means that the protein itself has more phosphate groups added to it, like phosphates being a post-translational modification and a regulatory effect. So, some proteins are active when they're phosphorylated in certain positions. Some proteins are inactive when they're phosphorylated in certain positions. In this case, you have a lot of extra amino acids in tau that are phosphorylated in AD. And this causes the microtubules to disassemble. So, normally tau keeps them, ordered and properly assembled. And when it's hyperphosphorylated, it can't do that anymore. The tau fibrillizes and aggregates into these neurofibrillary tangles. This is a really important point. So, how I was saying with the senile plaques, with the A beta plaques, they don't actually, the number of them doesn't actually correspond with progression or how bad the disease is. But for NFTs, neurofibrillary tangles, that's actually well correlated with the progression and the effect of AD. Okay, so what is this APOE that I was talking about? We're going to talk about that next. So, it's the E4 allele that's thought to add risk to people. And that is the APO stands for apolipoprotein E. So that's the name of the protein. And E4 is the particular allele. So we don't call it a mutation because it doesn't necessarily sort of affect the protein in such a way that it's disabled or something like that. I mean, mutations usually are things that cause disease, whereas the E4 allele would be considered a polymorphism. So everybody's genes, they can be slightly different from each other without causing any problems. They're not pathological changes. And so that's the case for E4. It adds risk, but it doesn't actually cause the disease. So what is APOE? It is a 299 amino acid protein. It's a primary lipid transporter. Sorry, it's a primary lipid transporter, and it's found, there's a lot of it in the brain. So the main alleles, so I said they were polymorphic, are E2, E3, and E4. They might be more alleles, but they're quite rare. So it is the most common inherited risk factor for AD. So remember me saying that familial AD is caused by like a mutation or a change in a particular gene, in one gene. So the thing is that the risk factors, they don't cause the disease. They simply, like, if you have the E4 allele, you're just that much more likely to get it. But it doesn't actually, like, if you have it, it doesn't cause the disease, if that makes sense. So the genome-wide association studies, that's what they're looking for. They're looking for things that might make it more likely for you to get a disorder. Whereas this one, the E4 allele of APOE, is so common. It was actually found before the genome-wide association studies. And so 15% of people with the sporadic version, this isn't the inherited version, this is just the one that, people in their family don't necessarily have Alzheimer's, so they haven't inherited it directly, but 15% of the people that have the sporadic version actually have the APOE4 allele. So it is quite an important inherited risk factor. So 40 to 80% of AD patients have at least one APOE4 allele, and if they have one allele of E4, the risk increase is 3 times. If they have 2 E4 alleles, so both copies are the E4 allele, the risk increase is 15 times. So that is quite an extensive increased risk. But like I was saying, this isn't true for some African ethnicities. The APOE4 does not add to the risk in people of African descent. And part of the theory around this is that it's possible that it seems that generally people of African origin have, or African ethnicity, have lower cholesterol levels, and that seems to add in to the risk. So the people that are most likely to develop Alzheimer's disease are people with E2, E4, E3, E4, and E4, E4. Interestingly, there's another APOE4 allele that is thought to be protective, and this is the APOE2. And so people with APOE2, and E2 and E3 actually are less likely to develop Alzheimer's. So it's a very interesting risk factor. What does APOE do? Well, it's thought that APOE might actually help the aggregation of A beta to form the amyloid plaques, and that the APOE4 might be slightly more likely to make the aggregates than APOE3 or APOE2. Okay, so now we come to the GWAS, the genome-wide association studies, looking for these genetic risk factors. So what does this really mean? So what it comes to is there are certain mutations that just outright cause Alzheimer's. And those are known as, Alzheimer's genes, essentially. There's a mutation in the gene, it causes Alzheimer's. People with the mutation get Alzheimer's disease. And so those are the ones that I've mentioned before, APP itself, which is the precursor to A beta, and then the presenilins, which are part of the gamma secretase. And so if you look at the code here, they're involved in APP metabolism. And those are quite rare. Remember, that only comes to 1% of people with AD, and that's all of them together. So what are the things that we're looking for in these genome-wide association studies? We are looking for the rest of these. So this is what we were talking about before. So APOE4 is adds such a risk that it's actually been identified before the GWAS studies. The GWAS studies, or GWAS. 2 copies cause quite between a medium and a high risk. 1 copy causes more of a between a medium and a low risk. And if you look along this axis, these are the rare things that you might find. So finding the mutations is rare. Having 2 copies is not that common. Having 1 copy of APOE4 actually is more common. And then all of these kinds of risk factors that have been identified in GWAS studies are much more frequently found in the population. So a lot of them can be quite common. However, the point is that each of them only adds a slight bit of risk to getting AD, and it's a combination of, actually, I haven't mentioned this before, but environmental factors as well as these kind of genetic risk factors that is thought to cause Alzheimer's disease. Though we did say about 70% is thought to be heritable, but it might be a lot of these factors rather than any of these factors that's doing it. So where do what are these, GWAS genes? I mean, what are the GWAS genes doing? Like, how do they fit into all of this? So I'm not going to go through this in detail, but just to say that, we have the amyloid cascade hypothesis, and the red, the fully red genes here are the ones that are mutations, and then you see all of these outline red genes. Those are ones that have been found in GWAS studies. And you can see that actually some of them might fit into the tau metabolism, some might be involved in blood-brain barrier clearance, some might be involved with microglia clearance. So it's thought that microglia can actually clear some of the plaques, clear some of the A beta, etc., and that might actually help. There's some actually that have to do with the beta secretase, and there are some others that generally just have to do with the APP metabolism. And there's a theory that's in this paper here, which says, maybe actually it's something to do with the focal adhesion complex. We didn't actually talk about that theory. It's another theory, yet again. And so the idea is that all of these factors are involved here. So these guys in gray are actually genetic risk factors. And actually, APP is obviously one that can cause a mutation. A mutation can cause AD. But and then some of these are involved in tau, and some of these are involved in APP. And again, if you look more generally, this is actually more about this particular paper's theory, which is around the focal adhesion complex. So they've tried to work in how this theory can cause Alzheimer's disease, and fit in all the different sort of genes that have been identified in these genome-wide identification association studies. So they're comparing the sort of current, which is actually the A-beta, the amyloid cascade, APP, A-beta oligomers, tau pathology, synapse and neurite dysfunction, and they've kind of put it into a core of focal adhesion, and you can have a look at the paper to find out details, but that's their basic model, where there are different things that can sort of lead into it, but it all together causes the disease. All right then. So I will say again, if you really want to find out, I mean, I went through these slides quickly, but it's just to say that part of what people are doing with the genome-wide association studies is looking at the different, looking at all these different theories and the pathways that we think are involved, and then trying to sit them into those pathways. So like, how do those new genes that have been identified as risk factors add in and sort of add information to what might be happening in Alzheimer's disease? So if you're really interested in this, I'd recommend reading this paper to find out. I found it quite interesting. All right. So just as a final summary, we know some of these things are happening. Exactly how they cause the neurons to die, we don't know. But what we have is APP in normal state. It gets cut by the alpha secretase protease and the gamma secretase, and you get, these component parts. Tau binds and stabilizes the microtubules. In Alzheimer's disease, you get the beta secretase cutting APP first, and then the gamma secretase, and that leaves you with the A-beta peptide. You get oligomers, A-beta misfolding, aggregation, amyloid plaques. And see, this is actually one of the connections that's not really clear, which I may not have stated so far, but essentially when you have another problem with the amyloid cascade hypothesis is that it's not actually clear how the amyloid plaques lead to anything happening with tau. But we do know that these plaques probably, the A-beta somehow increases reactive oxygen species and somehow activates kinases. So kinases are the proteins, again, that add phosphorylation, who phosphorylate, add phosphate groups to proteins. And so tau gets increased phosphate groups. That causes it to aggregate and not bind to the microtubules anymore. The microtubules destabilize, and the tau gets sequestered in the neurofibrillary tangles. And again, there's some wonder if this is actually protective for the brain, and that's why it happens. Okay, so that is our summary of Alzheimer's disease, and I'm going to stop here and then start a new video talking about sort of the first half of the Parkinson's disease material. Thank you very much.